This recording is going to cover the anatomy of skeletal muscle. This is needed in order to understand the physiology of how skeletal muscle contracts. So this first recording is just going to discuss the basic anatomy of a skeletal muscle. So we're going to start looking at entire skeletal muscle. So just think about your hamstring muscles, including biceps femoris, semitendinosus, semimembranosus. So this picture right here would represent, say, the biceps femoris. Now, if you look at the muscle, or the muscles in the back of the thigh, they're kind of separated from each other. And one of the things that kind of helps to separate them from each other is connective tissue. And so we're going to be looking at, in the anatomy of the muscle, we have to discuss some connective tissues also. So the skeletal muscle itself is surrounded by a connective tissue layer that's referred to as the epimysium. It is composed of primarily of dense irregular connective tissue, some areolar tissue. And what you'll notice is, if you look at the picture, some of the things that you see associated with it is you see this yellow. This is a nerve, and you see some blood vessels. And so besides kind of separating muscles from each other, the epimysium is going to help to support nerves and blood vessels that will be associated with the muscle. You need the nerves in order to tell skeletal muscle to contract. You have blood vessels to deliver oxygen and nutrients, and then veins to be able to take away deoxygenated blood and waste products from the muscle. And also the epimysium is connected or is associated with the tendons and the flat sheet-like tendons referred to as aponeuroses. So when a muscle contracts, and we'll be looking at the smallest component of the muscle, it has to pull on these connective tissues, and eventually that connective tissue associated with the tendons. So if you want to be able to move the bone, you have to kind of pull on the tendon. So if we kind of go back to looking at that skeletal muscle, it's made up of various components. So if you look at each one of these that you see here in this skeletal muscle, that is referred to as a muscle fascicle. So this is a muscle fascicle. So if we kind of fill in this chart, though, he says the top one says surrounded by epimysium, it contains the muscle fascicles. Now the muscle fascicles contains muscle fibers, which is what we're going to look at next. So over here, this is a muscle fiber. So we'll look at that in a moment. The muscle fascicle, like when you learned about nervous system, you learned about nerve fascicles. So you say, I got a nerve, it consists of a bunch of nerve fascicles, and the nerve fascicles were bundles of axons. So kind of it's synonymous when you look at the nervous system is we have a muscle that has a connective tissue layer associated, we call the epimysium. That's composed up of muscle fascicles, and the muscle fascicles are surrounded by connective tissue that is referred to as the perimysium. And again, what you'll notice is you see blood vessels and you see nerves, because we're going kind of deep into this muscle, and you have to kind of you can't just, an entire skeletal muscle doesn't necessarily contract, as some of the components of it will contract. Okay, so not always will you have every component of a skeletal muscle contracting. So if we look at the muscle fascicle, zoom in on that, the muscle fascicle is comprised up of muscle fibers. And the individual muscle fiber, which you kind of see right here, the wrapper on it, that's surrounded by endomysium. Again, the endomysium will be supporting things like the capillaries, the smallest component of the blood vessels. It'll support some of the nerve endings that'll eventually tell the skeletal muscle fiber to contract. Now, the skeletal muscle fiber is actually a skeletal muscle cell. So it's, when you think of a cell, you think of a nucleus, you think of mitochondria, you think of your various organelles. Same applies here. And when we look at skeletal muscle, we're going to have some slightly different names for some of the organelles. So the skeletal muscle fiber is actually contains a number of myofibrils. And kind of a reminder, myo means muscle. You see mes here, that means muscle. Okay, so you'll see kind of some of the terminology. Now the myofibrils, which you see right here, these consist of sarcomeres, which actually the sarcomeres consist of what we refer to as myofilaments. The myofilaments that we'll discuss will be actin and myosin. Actin is the thin filament, myosin is the thick filament. Now if you look at the myofibrils have, you can kind of see in the blue is actually a organelle called the sarcoplasmic reticulum. We're going to see that in the next slide. The yellow, which you see here, is actually referred to as a T-tubule, and I'll show you again in the next slide. Now, when we want muscle to contract, actually the sarcomeres are the contractile elements of a skeletal muscle fiber. When what happens is during muscle contraction, all the sarcomeres within a muscle, or we should say within a myofiber or myofibril, will contract. What we may need more muscle fibers to contract in order to generate enough tension in order to move something. And so we may need more and more muscle fibers. And at some point, if you need all of them, you'll have the entire skeletal muscle contracting, but we're going to be seeing that very shortly. And we're going to be looking at this sarcomere a little bit more in depth. So here, if we're looking at the muscle fiber and the myofibrils here, the muscle fiber is that muscle cell. And you learned in biology that muscle cells are surrounded by a plasma membrane. Well, this is a muscle cell, but the muscle fiber is the cell membrane is actually referred to as a sarcolemma. Sarco is a term that literally means flesh, so meat, to eat meat, you eat in the flesh of an animal. Lemma is a term that means husk, so think it's the husk like a corn cup. So the sarcolemma is the plasma membrane. And if we look at the bottom picture, you see that sarcolemma again here. But what you'll also notice is there's these structures that are invaginations of the sarcolemma, and it's the T-tubules. And the T-tubules are what you see in yellow. The T-tubules are also referred to as transverse tubules, or you can abbreviate as the letter T, tubules. They're invaginations of the sarcolemma. They're going to serve a purpose, is when an action potential is generated along the sarcolemma. It will be propagated along the sarcolemma, but it needs to get down deep into muscle. The muscles are fairly big, and so we need to propagate the action potential deep down into the muscle in the transverse tubules. That's going to be their function. Now, the over here, what you see in the blue, that is referred to as the sarcoplasmic reticulum. And actually, where this line is going to, it's actually going to what we refer to as the terminal cisterna. This is singular with the A, plural with an E. The terminal cisterna are like the endings. Some people refer to them as lateral sacs of the sarcoplasmic reticulum. The sarcoplasmic reticulum sounds like endoplasmic reticulum because it is. But the sarcoplasmic reticulum in skeletal muscle, you notice there's sarco as part of this term again. The sarcoplasmic reticulum contains a lot of calcium ions. This is a membrane-bound organelle, and we're going to hold a whole bunch of calcium inside it. When we need it, which we will need for muscle contraction, the sarcoplasmic reticulum will release it into the sarcoplasm. The sarcoplasm is the term for the cytoplasm of that skeletal muscle. Now, the terminal cisterna, you see one here, you see one here. That, along with one of the T-tubules, is referred to as a triad. So it has three components to it. The triads are important components of skeletal muscle. If you look carefully, is you notice that the triads are positioned over these areas right here. And those areas, and you can kind of see it right there too, is referred to as the zone of overlap. This is a region where actin and myosin, which are the myofilaments, are overlapping. So on the picture of the myofibril, which they've pulled out to show you, this one here is myosin, the thick filament. This one is actin, the thin filament. These two structures need to interact for muscle to contract. And I had mentioned that calcium is going to be very important. So we have positioned these triads over the areas of overlap so we can make sure we can get that calcium to where we need. And we're going to show you where that calcium is needed in order to get muscle contraction. Also in the picture, what you'll notice, they've labeled the mitochondria, which you see right here. So mitochondria produce ATP, and you have to use aerobic means. In mitochondria, you need oxygen. So this produces a lot of ATP needed for muscle contraction. But skeletal muscle can also generate ATP using glycolysis, and you don't need mitochondria for that. But we'll talk about energy generating pathways a little bit later. So these are some of the major components of skeletal muscle. Now, one thing I forgot to mention is what you'll see here and here and here and here. These are the nuclei. In skeletal muscle is multinucleated. A skeletal muscle cell is actually really, really large, a lot larger than cardiac muscle or smooth muscle. And you need a lot of nuclei, and they're peripherally located. They're on the outsides. You need a lot of nuclei because there's a lot of turnover of proteins like actin and myosin. And so the nuclei have that information in order for you to produce various proteins. The proteins obviously are going to be produced associated with the ribosomes, which either is associated free in the cytoplasm or associated with endoplasmic reticulum as the roughy are. But the nuclei have the information in order for you to be able to produce those proteins. So there's a lot of turnover in a skeletal muscle cell, so you need a lot of nuclei. Now also what you'll notice on the top picture is the striations. So when you looked at when you did histology, you should have noticed that skeletal muscle was striated. And the reason why it appears striated is because of a regular arrangement of the actin and myosin, and we're going to see that very shortly. Smooth muscle does not have striations. Now, smooth muscle does have actin and myosin, but there's not a regular arrangement of it, so it does not appear striated. Cardiac muscle also appears striated because it has a regular arrangement of actin and myosin. Now, if we look here, if we look at that myofibril and look it up close, we're going to look at the sarcomere. So that sarcomere I had mentioned was the contractile element that we see in muscle. Now, what we have, the sarcomere extends from, we see the Z line to this is another Z line here. So that's the sarcomere, and you've got lots of these within one muscle or within one myofibril. And what we'll see is in the kind of the purple represents the thick filament, which is again myosin, and the thin filament represents actin. And you see kind of this regular arrangement of actin and myosin. Now I want to talk about some of these components of the sarcomere, some of these bands and these zones. Now the M line, what you see how you associate M line with the middle, the actin you see is associated with the M line. The Z line, some people call it a Z disc, you'll see actin is bound to it. So actin is bound to the Z line. Now, what you'll also notice in the green is this protein called titin. Now, titin is a very large protein, and it has some elastic qualities to it. One of the things, it does a number of different things, but one of the things it does is you see it's attached to myosin, so it allows kind of keeping this regular arrangement. So kind of keeps everything in the right configuration. So it's attached to myosin, but also after muscle contracts, you have to go back to its original position. And because it has elastic qualities to it, it allows things to go back to its original position. Now, what you'll notice is you'll have this region in the middle, which you refer to as the H zone. Now, the H zone, what you'll notice is only has myosin. There's no actin associated with the H zone. If we look at the A band corresponds to the length of your myosin. Now, the A band consists of both myosin and actin, includes this region of the zone of overlap, which I had mentioned earlier, that region where actin and myosin are actually overlapping, but not necessarily interacting yet. So we'll have to talk about that. Now, also the I band. The I band is on either side of the Z line. So the I band only has actin. Now, I do want to mention, this is what we're seeing, how things look at rest. The sarcomere is at rest. But when muscle contracts, we're going to see some changes in the lengths of some of these bands or zones. What actually happens is actin is going to be sliding in this direction toward the M line. And what happens is the size of the H zone decreases. The distance of the I band decreases, but the A band stays the same. The A band stays the same because it's the length of the myosin. We're not going to change the length of the myosin. But what will happen is during that contraction, that zone of overlap is actually going to increase because actin is sliding towards the M line, so they have more of a region of overlap. But what it creates too is the interaction between actin and myosin, and we're going to show you very shortly how they interact. Now, what I want to do is look at myosin and actin a little bit closer and kind of look at what they're comprised of. So we're going to look at myosin first. Myosin, I had said, was the thick filament. And what you'll notice is that the myosin molecule actually has these little heads, which you see here. And the heads have a spot, a binding site for actin. And the interaction between actin and myosin is important during muscle contraction. It has a binding spot for it, but it also has an enzyme called myosin ATPase. ATPase hydrolyzes ATP into inorganic phosphate and ADP. Now, the ATP and its hydrolysis is going to be important in muscle contraction. Now, what you're going to notice on the picture is that down here at the bottom, I'm going to erase it here again, you see that the myosin head can move positions because there's a little hinge that we have here, a little hinge joint. So what we'll see is during muscle contraction that you'll have that head pivoting, the head pivoting, which is going to be important as we're trying to grab onto actin and kind of move actin along myosin. So we're going to see that very shortly. So also what we're going to do is let's look at the thin filament or actin. Now, actin has a lot more associated with it. Actin has these regulatory molecules called tropomyosin and troponin complex. But let's just look at the actin itself. On the picture, it's those beads. And actin is actually composed up of this F-actin strand, or we call filamentous actin, which are like, I equate it to like strings of pearls. So you have two strings of pearls and then they're coiled around each other. Each of the little beads or the pearls are referred to as G-actin. Okay, so that's the actin molecule. But what you'll notice is you're seeing these other things associated. We're not going to look at nebulin. I'm not going to talk about nebulin. But I want to talk about troponin complex and tropomyosin because they're going to be very important in regulating the interaction between actin and myosin. So if we zoom in on troponin complex, or I should say we zoom in on actin, let's look at the troponin complex first. Now the troponin complex you see right here, here, and here, it has three components. It's a complex. It has these three subunits. It has I, C, and T. Now, I do actually got to go and mention tropomyosin before I tell you about I, C, and T and troponin. Tropomyosin, you'll see, is this kind of strand, it's in gray on the picture, that's coiling, turning around the actin molecule. When calcium levels in the sarcoplasm are low, it actually physically covers a myosin binding site, which you kind of see over here. Okay, so here it's being blocked. So myosin could not interact with actin. But what we want to do is we want to move tropomyosin out of the way when we want muscle to contract. In order for muscle to contract, actin and myosin have to interact. So how does this happen? Well, this is where troponin complex comes in. So you see troponin complex periodically arranged along that tropomyosin molecule. But what we have is we have three components to it. Now, the troponin T is the part of troponin complex that has a hold of tropomyosin. Okay, so I got it like I got tropomyosin, got this rope in my hand. The troponin I is the part of troponin complex that's holding on to actin, because this is a regulatory molecule associated with actin. So I'm kind of holding the troponin complex in place with using I holding on to actin, T is holding on to tropomyosin. Now troponin C, and the way to remember it, troponin C has a C in it, it binds to calcium. So when calcium levels are high enough in the sarcoplasm, calcium binds to troponin C and it causes a conformational change, which means it's a change in its shape. And what happens when it changes its shape, it's going to physically move tropomyosin out of the way. So you'll notice at the bottom, the tropomyosin got moved and now the myosin binding sites are exposed. So now we'll be able to have actin and myosin interact. But as a warning is you'll need other things to be involved in order for myosin and actin to interact. But now we've at least moved tropomyosin out of the way in order for actin and myosin to interact. So this is the end of looking at the anatomy of skeletal muscle and the major components of the skeletal muscle. Next recording is going to cover some of the physiology of muscle contraction, including how did we get that calcium to be released into the sarcoplasm? How does actin and myosin interact? How is the ATP involved in this interaction? So we'll look at that in the next recording.